Hello, everyone. I'm Ginny Lobel with Cello Health BioConsulting, which is the relatively new name of our legacy company, Defined Health. Welcome to today's Insight Series webinar, Better Than A-Beta, Looking Ahead in the Battle Against Alzheimer's Disease. Today's panel will be moderated by Joel Sandler, Associate Principal of Cello Health BioConsulting, and our esteemed panelists today are Howard Phillett, Founding Executive Director and CSO of the Alzheimer's Drug Discovery Foundation, Ken Mock, President and CEO of Cognition Therapeutics, and Kiran Reddy, President and CEO of Praxis Precision Medicine and Venture Partner at Blackstone Life Sciences. For those of you who are not familiar with us, Cello Health BioConsulting, as mentioned, formerly Defined Health, is a Cello Health business. Cello Health is comprised of a group of subsidiary companies offering strategic commercial advisory services, valuation, and risk mitigation strategies across the development spectrum from preclinical through phase 4 lifecycle management, repurposing, reformulation, and pre, peri, and post-launch competitive analysis for life sciences companies globally. Our team at Cello Health BioConsulting is scientifically deep, and we assist clients with early development and risk mitigation strategies, including indication prioritization and sequencing for their compounds, platform technologies, and portfolios. As well, we conduct search and evaluation for portfolio expansion at all stages of development, and we provide independent and well-regarded forecasting in advance of investment and partnering events. Before we get started with today's discussion, I'd like to call your attention to our 30th Annual Cancer Progress Conference to be held in New York on May 7th and 8th, 2019, of course. Please take a photo while it's up of the QR code on the slide for more information about this unique and fantastic event. To learn more about us, give us a call, send us an email, or visit our website at cellohealthbioconsulting.com. Please note that an audio, visual, and PDF version of the slides from today's event will be available on our website within the next few days, and you'll be notified when they're posted. During today's discussion, you may send any questions you may have via the webinar system in real time, and we'll try to get to them either during the discussion today or at the end of the session, or we will respond to you directly post the event. So thank you for listening to my brief introduction. Please enjoy this edition of our Insight Series briefings, and I will now turn the mic over to Joel Sandler.
Thank you, Ginny, and thank you, everyone, for joining us. Thank you, of course, to our panelists who Ginny briefly introduced, and we'll be giving them plenty of opportunity to voice their opinions about where we are today and where we see the field going. Such a relevant topic. Very glad that everyone could join us on this Wednesday morning or afternoon, as the case may be. So it's impossible to overstate the issue with Alzheimer's disease. It's a pressing need that's only going to get worse in terms of cost, in terms of prevalence, in terms of how deadly it is, and in terms of how insidious it is. And despite all of the clinical risk, and we can talk to that, there's plenty of commercial potential in this space, and that's underscored by two symptomatic therapies that have gone on to become multi-blockbusters. The failures have taken their toll. No one, I assume anyone on the line is no stranger to the fact that we've had myriad clinical failures, and really nothing has panned out and translated from phase 2 success to phase 3. Most recently, of course, as you've seen in the press and are well aware of, Biogen and Eisai's aducanumab failed. That was perhaps the last hope for many of us who were hoping for monoclonal antibodies to address amyloid beta specifically as a therapeutic modality to address unmet needs. Some regarded this as vindication, others regarded it as a surprise, or at least as can be gleaned from the hit to Biogen's share price and market cap, an impact that wasn't altogether expected. That said, there's plenty of investment dollars, plenty of deal flow in neurology in general, but a lot of this, specifically if we were to break it down, which we haven't done here, is owned specifically by neurodegeneration, and within that, Alzheimer's plays an outsized role in terms of where the money is flowing. The government, at least in the U.S., seems to be on board, both in terms of funding and at least in terms of voicing the opportunity for companies to present surrogate endpoints as with the proviso that they are validated, and we can talk about that with our panelists, what validation means. That at least there is a willingness to accept things other than cognitive function and other aspects that perhaps have set too high a bar for clinical programs up until now. Of course, there are companies that are going to continue to throw good money after bad, and maybe we'll see some glimmers of hope from future studies in anti-amyloid antibodies. But that said, I think there's plenty of additional waves of innovation, including approaches that are trying to address amyloid beta in terms of its pathological implications, not necessarily the plaques, but perhaps other components of A beta pathology, and looking upstream and looking at different parallel pathways that might collectively help chip away at the issue. So my questions for my panelists, and we have a short amount of time, so we will do our best to get through all of these, but the questions I have are kind of threefold, or can be divided into three categories. One is, what have we learned from aducanumab and other anti-A beta failures up until now? Are we still, is the amyloid hypothesis still alive? And if so, are we forced to reckon with a modified version of it? Are we looking at the right endpoints in our trials? I mentioned surrogate endpoints. I think a big push in the field going forward has to be diagnostics as a companion, or at least a means to de-risk some of the clinical therapeutic candidates. And where are we today in terms of perception, in terms of what partners think, what investors think? And we have, I think, a great panel to answer those questions. I think more to the point, and I suspect what will be relevant to our listeners, is where do we see the next waves of innovation coming from? Whether it's therapeutic mechanisms, targeting immunology, glial cells, astrocytes, various gene therapy approaches, infectious disease, and the components that they may, the role that may play in pathology, gene therapy, cell therapy, as well as other ways to drug the role being played by amyloid beta and tau and components that have been on our radar up until now, but perhaps the modalities that we've been throwing at them haven't been optimal. What role, as I asked, should diagnostics play? I think that's a big question for everyone. And what will it take for this field to really heat up in a way that we saw in circa 2013 and it's still present and still going in the oncology space, and more recently with gene therapies and rare diseases? And then I think another question is that of what I collectively refer to as macro factors, headwinds, tailwinds, economic incentives, pricing concerns, should we get to a point where we have a therapy that's relevant for a large proportion of prevalent Alzheimer's. Who are the dominant investors? Are there venture arms, which I'm hoping Karen can speak to, as well as charitable groups, which I'm sure Howard can speak to, and what are the complementary roles that each of them can play in terms of the needs that are being addressed? And what role can regulators play, and where are we with that, and what are ultimately the takeaways for the companies, the innovator companies, the investors, the business development groups at large pharmas that might be in the market for acquiring innovative approaches from these companies. So with that, I'd like to start with the question, and maybe I'll put it to Ken, who is our representative from biotech, to the question of what does the aducanumab failure, most recent failure, mean to the field, both from a technical perspective in terms of how to drug the disease, and also from a perceptual perspective in terms of the reception you get nowadays in the last couple of weeks relative to before this from investors, from partners. So Ken?
All right. Can you hear me? Just confirming.
Yes, you sound good.
Good. Great. So I think that the evidence suggests that the monoclonal antibody approach to, you might want to leave up the questions, by the way. I think that's a real, if you don't mind, that'll be the best place to be. The monoclonal antibody approach to the amyloid hypothesis, I think, has some significant nails in its coffin, both from the standpoint of looking at plaques and fibrils and looking at A-beta monomers as well. As Howard and I had this discussion, that aducanumab, to a very large extent, did what its target engagement approach was, which was to reduce amyloid plaques, but it didn't correlate with a clinical outcome, and that may go to one of your later questions. So I think from that perspective, we see that issue reaching the end of its scientific pathway, or at least for now. On the other side of life, on the overall question of what does this mean for the amyloid hypothesis, I think it depends on how you look at what the role is of A-beta. From the perspective of cognition therapeutics, we're looking at A-beta from the standpoint of A-beta oligomers and the toxicity of A-beta oligomers as it relates to direct interaction with synapses. So not to get too far ahead of your question, but every genetic mutation associated with Alzheimer's points back to A-beta, and in fact, the one genetic mutation that's positive points back to A-beta. So there's something in there, and we look at it from a small molecule perspective and really look at our approach as toxic oligomers interacting with synapses and protecting synapses. That's side A of your question. Side B of your question, relating to the perceptions, I had been concerned that a Biogen failure would further depress an already depressed field. To my surprise and delight, we have seen almost more interest and people trying to figure out where to go next. This is one of the largest remaining, if not the largest remaining, undealt with medical emergency in our society. I look at it as a societal tsunami. We must do things, and people are not willing to give up on this, and that has been fabulous to see, and frankly reflected in discussions that we've had at Cognition Therapeutics, even within the past couple of weeks. So to be pithy, I'll stop there and let other people step in.
Yeah. Howard, maybe I'll put it to you. So aducanumab's only the latest in a long string of failures targeting or trying to develop antibodies against amyloid beta. There are also small molecule approaches against enzymes that are involved in the creation of these plaques. Do you believe that there's anything salvageable putting aside the amyloid hypothesis and getting more specific, is there anything from these drugs left that hasn't already been tried, or do you believe that it's time to really kind of look forward and move on?
Yeah, thanks. I think it's pretty clear from multiple failures with gamma secretase inhibitors, beta secretase inhibitors, and the antibodies against the various domains of the A beta molecule, the N terminus, C terminus, the mid terminus, all different kinds of immunochemistry, that these drugs are not improving clinical symptoms. But I think that the aducanumab trial was a landmark trial in our field. We've only been doing clinical trials really in our field for about 30 years. And if you consider that, cancer and all the other spaces have been doing clinical trials for 60, 70, 80 years, we've had a lot to learn. And of course, biomarkers play a critical role. The aducanumab trial was sort of a landmark trial because it was really the first time that a clinical trial for Alzheimer's disease required a PET amyloid scan to get into the trial. It required that people getting into the trial actually had the disease under study and actually had the target under study. And then it used that same biomarker, the amyloid scan, which ADDF helped to develop actually, as an outcome measure in the trial. And it showed, as Ken mentioned, that there was target engagement and that the drug worked according to the way it was designed to work. And the studies with the amyloid PET scan have shown, when Lilly used it in the solanezumab trial, that up to 35% of people entering into that trial and deemed appropriate by dementia experts at clinical trial sites didn't have amyloid deposits in their brain, and they didn't probably have Alzheimer's disease. So this was the first really rigorous trial in our space, and it was actually the first time that, I think as Ken alluded to, that we had a very rigorous approach to the hypothesis that these plaques particularly played a role in cognitive decline in patients with MCI and mild dementia. And we got an answer. And the answer appears, we haven't seen all the data yet, but appears to be that you can remove these plaques, at least based on what we see on PET scans, but it doesn't really affect cognition, cognitive decline over time. So what does this mean? Well, I think it's clear that I think that's clear when we also factor in the beta secretase failures, that maybe amyloid is not the right target. The remaining issues are, of course, are we treating early enough? As a clinician, I know that when I see people with MCI or even mild dementia, these are functioning human beings. People with MCI have very little functional impairment. It's almost by definition that they don't. If people with MCI do have significant functional impairment, then they should rightfully be called mild dementia patients. And what that tells me is that people with MCI have lots of salvageable neurons left because they're still functioning very well in the community as human beings. So are there neurons left to save at this age of MCI? Clearly, yes. Do we need to go even earlier than MCI to find the sweet spot for treatment? I think that's kind of the hypothesis, and it's based on the fact that amyloid accumulation, again, we know this from this revolutionary PET scan that we have for the first time. We can show that people start accumulating amyloid in their brain 20 years before they become symptomatic. And so the theory is, well, maybe we can go into a primary prevention trial, or if you want to call it secondary prevention, a trial where people only have the pathology, they have no symptoms, and it's going to work in that case where it didn't work in people who are symptomatic. And as somebody said recently, there's no disease that anybody really knows of where, that kind of prevention works and the treatment trials didn't work. But nevertheless, the remaining question really is, given the timing of amyloid deposition being pre-symptomatic, and not correlating with cognition, by the way, so that there's another bias out there that because the beta amyloid accumulation doesn't associate with the progression of clinical symptoms, whereas tau imaging and tau tangles pathology does, is this really going to work? And I'm kind of skeptical that these prevention trials are going to show us anything. I think part of the reason is that if we look at the biology of aging, amyloid, beta amyloid is a normal protein that has some function at the synapse. We don't know exactly what that is, and it becomes misfolded. And we know that in the aging brain, there are many misfolded proteins. There was recently a study that showed that the plaques by mass spec have up to 4,000 proteins in them, 40 of which accumulate in significant amounts. So these plaques are kind of reservoirs of misfolded proteins that the body seems to be able to want to ward off into a special kind of pathology. And so the question is, if you target with an antibody one of these molecules out of the many that are misfolding and depositing in plaques, is that going to have a clinical effect? Or by the same token, the many misfolded proteins that are probably accumulating in the synapse, like tau, which seems to be misfolded and maybe transmitting, skipping synapses. And my point in making this is that maybe it's the wrong target. Maybe we need to target things like autophagy and proteostasis so that we can have a more parsimonious approach to these misfolded proteins.
Great. And Karen, I want to, Howard, I think that's a great segue into some of the next wave things, but I just want to stick with the question of A-beta antibodies for a few more minutes, and I'll put it to Karen, and maybe this does become a good transition to next wave. There's a question from one of our audience members that came in virtually about whether aducanumab and other antibodies against A-beta actually cross the blood-brain barrier, and how the A-beta plaques are processed and disposed of, assuming that the antibody does get in there and form a complex with the pathological target. So this, to me, begs the question of really gets at the question of separating the rationale for this approach and the logistics of it. And I don't know if we've solved that question or not. Presumably, some of aducanumab gets past the blood-brain barrier. You have companies now like Voyager with vectorized AAV capsids that are getting antibodies across the blood-brain barrier, only to be expressed once across the blood-brain barrier. So that might help get at that question of whether, if we had an antibody in the brain at sufficient levels, and we may or may not know if that's been accomplished to date, whether such an approach would be efficacious. So Karen, can I put that question to you? Do you have any thoughts about that?
Sure. So yeah, happy to try to answer that question. In terms of monoclonal antibodies, blood-brain barrier permeation, we generally know in the field, we're getting 0.1%, across the blood-brain barrier. So that, clearly is a problem in terms of getting sufficient, concentration of antibody into the brain, though we can do it, in that, if we are flood the peripheral circulation, we can get 0.1% and can achieve what therapeutic levels we seek to ascertain, in some cases, and in the case of aducanumab and the more recent A-beta antibodies, I think, as Howard nicely described, there has been proof of target engagement. One of the things that Biogen did very nicely was show plaque reduction. So I don't think the aducanumab failure was an issue of not getting sufficient antibody into the brain. It achieved its therapeutic purpose. Again, as Howard nicely went through, it reduced plaque. I think so I don't think that that getting higher concentrations of A-beta antibodies in the brain would lead to a different result. If I could just briefly describe an analogy of cardiovascular disease and atherosclerosis, which is often compared to what might be, happening in terms of Alzheimer's disease and pathophysiology. I mean, it is somewhat, disconcerting that all of these various A-beta approaches have not worked. We were also crude in our understanding of atherosclerosis and cardiovascular disease, yet, approaches, that were, lowering lipids in different ways or lowering inflammation in different ways with, it would be aspirin or various LDL lowering mechanisms. They worked. They worked to decrease cardiovascular disease, and we didn't have nuanced understandings. But here, there was certainly quite a bit of support that A beta and plaques, likely have a role in pathophysiology. Yet, we've seen with these various trials that they have not, had real clinical benefit. So we are left with a more complex situation in Alzheimer's disease than we are in cardiovascular disease. I guess to provide my opinion on, is there a nail in the coffin on the A beta hypothesis? I guess I would say that I'm not quite ready yet to put that nail in the coffin. It may be that we don't have in A beta a solution set for MCI for early AD in this broad population that we might really need to understand. Are there subsets of patients where in overall Alzheimer's disease and MCI and in early AD where you could find that plaque reduction would be meaningful to improving their clinical outcomes? That clearly, when you take all comers, that was not the case. But we have certainly seen throughout medicine that there are situations that, as we better understand what might be the pathological drivers for different patients with Alzheimer's disease, these are not all the same patients. There is likely complexity here, and perhaps either A beta approaches, A beta plaque reduction, or A beta production approaches will be useful for certain subsets of patients.
Yeah. Okay, great. So let's go back to, I guess, as I said, this is a nice transition to thinking about next wave approaches. And by the way, we have a lot of questions coming in. They're all great questions. If we don't get to them in the course of this discussion, we will be sure to email you guys afterwards. So let me go back to Ken, because I think this question about A beta being the right target, the wrong target, and what the implications are. I know with cognition, Ken, you've been working hard to develop your lead program, which was capable of displacing a soluble A beta oligomer to neurons. The implications there are, one, that I believe, and you can tell me, one, that the overall target, or I should say the overall molecule is correct, but the specific target or piece of A beta may not be. And two, I think more intriguingly, and I've discussed this with you in the past, is the idea that the soluble oligomers are binding directly to the neurons and the synapses. And this gets into the question of synaptic resilience as a target, and coupling that with different biomarkers, whether it's FDG PET or other things that you guys are looking at, says to me that maybe there's a target here that doesn't need to be abrogated 20 years before the patient is symptomatic. What do you think about that, Ken? And to the extent possible, maybe broaden the question and the topic to the field at large, not just to your specific programs.
I'll try the second part, but I'm of course deep into the first part. I mean, I think the question that begs is, what's the target and why haven't things worked in the past? And there's evidence from historical clinical trials that, as Howard mentioned, A beta monomers are necessary in some way for synaptic function. And there's also a lot of evidence that A beta oligomers and plaques are basically the low energy state location or form, if you will, for how the body stores A beta monomers when there's either an overproduction or, more specifically, as we age, there's a decline in clearance of A beta monomers. So that could be why everything we've done in those two particular areas has not had a therapeutic benefit that's discernible. The A beta oligomer form, you know, and just moving to this for a second, and I'll try and get more general. You know, see, we have evidence that binds specifically to a site on the synapses, and if that's the toxic entity, the A beta oligomer, and the toxicity that is what we call Alzheimer's disease occurs from this interaction of a toxin with a receptor ligand, and that you can do something from a drug perspective to displace those oligomers and restore synaptic function, then that's a potentially important thing. Just look at it by preventing end organ damage. Is the brain an end organ, and can you prevent the damage, and how do you do that? And that's the approach that cognition has taken with synaptic protection, if you will. You know, and then what's downstream and upstream from that is really your broader question. I'm not as much of a detailed specialist in all of that, to be blunt, and you know, I think that's something you can throw to Howard or Kieran. I will say that biomarkers of damage, I don't believe you can do these long-term clinical trials and move earlier and earlier, not necessarily from the standpoint of they might not work, but rather that they're economically unfeasible. It takes too long. You know, I used to run an anti-aging company, and the joke was, Eureka, we've cured aging. The only problem is it'll take forever to prove it. As these trials get longer and longer and require larger and larger patient populations to get a p-value, they just become unfeasible. So, you know, that may be our complexity. And I also believe that we have a problem with endpoints. And I don't believe that everybody who's gone into the clinic with a new drug in Alzheimer's is wrong about the activity they've seen early in the drug's development. But I think the current endpoints are imprecise and variable and subjective. And so we're going to need things. Howard's got the Alzheimer's Drug Discovery Foundation has a major effort in biomarkers. Many people do. So between protecting the synapse or protecting downstream pathologies, which other companies are doing, and thinking about biomarkers, be that our particular primary thing of actually being able to count synapses and potentially protect synapses, or look at biomarkers of synaptic damage, I think this whole field could change in a relatively short period of time. We're certainly hopeful of that. So maybe, you know, for your broader question, you can turn it to my my panelist colleagues.
Okay, yeah, and I'll do just that. So Howard, I think of biomarkers broadly as, you know, being positioned to identify patients earlier, which is powerful, but as Ken says, leads to questions about actionability. Biomarkers can also be used to stratify and potentially enrich patients for a particular therapeutic approach. And biomarkers can be used to monitor the efficacy of therapies in real time in such a way that potentially forms a bridge between the therapy, the administration of the therapy, and the cognitive and functional endpoints, which to date have set a pretty high bar in such a heterogeneous patient population. So it strikes me that the Alzheimer's Drug Discovery Fund, which has drug discovery in the name, is so heavily invested in biomarkers, and I don't think that's a mistake. I think these biomarkers are part and parcel to de-risking and ultimately making therapeutics viable and successful. But I'd love to hear your thoughts about where you see things going in terms of application and positioning of biomarkers as a means to unlock the full potential of therapeutic candidates.
Sure. Well, let me give you a little perspective. Where do you think we'd be in heart disease today if we didn't have a blood test for cholesterol? Where do you think we'd be in oncology today if we didn't have all these genetic markers of subtypes, so that we're not looking at lung tumors on a chest x-ray anymore? We're looking at liquid biopsies. You know, biomarkers are critical. Where would we be in diabetes if we didn't have a hemoglobin A1C? Where would we be in hypertension if we didn't have a blood pressure cuff? We're going to have a blood test for Alzheimer's disease, I think, commercially available in about 3 years. That's why we formed the Diagnostic Accelerator with Bill Gates and Jeff and Mackenzie Bezos and David Dolby and some other prominent philanthropists. We have a $50 million fund. And the timing is right for several reasons. There are many biomarkers. We put out an RFP worldwide for biomarkers, for fluid biomarkers, basically blood, back earlier in the year, and we got over 300 ideas from 26 countries. So what I'm saying is that the panoply of biomarkers that are out there beyond plasma A beta are enormous. We may have a plasma tau test. We're going to have plasma inflammatory tests. We're going to have microRNA tests and even digital tests that'll help us. And we know even the FDA recently came out with the new guidelines. The whole point of the new guidelines was a regulatory perspective on how biomarkers will be used in regulatory approvals in these earlier stages of disease where we only have biomarker positive patients who are not clinically affected but have the pathology, as we talked about earlier. And if you think about the cost of trials right now, one clinical trial in Alzheimer's disease in phase 3 costs $300 to $400 million. That's not a lot of shots on goal for even a big pharma company. So they've had to really focus on what has been thought in the field to be the low-hanging fruit. You can't take a lot of shots on goal when each shot is costing you $300 to $400 million. But half of a phase 3 trial involves recruitment and enrollment costs. And part of the recruitment cost is getting those screens done to make sure the patients have the disease of interest in their brain, namely, either beta amyloid PET scanning or CSF, both of which have either cost or invasive problems in helping in getting patients to enroll. So if we had a blood test, those screening costs in the community would be markedly decreased. And by having a blood test out there for Alzheimer's in the community, we'd also have much greater cohorts of people who had biomarker-defined disease in the community, who would then be available to enroll in these clinical studies. So, for example, like in other diseases, less than 5% of people with Alzheimer's disease actually enroll in clinical trials today. So, as we think forward, what we're going to have, it's going to be, in my view, like cancer. There are many mechanisms. This is a disease of aging, just like cancer and heart disease and so on. And so we're going to have a lot of pathways. And maybe beta amyloid therapies will be one of a combination therapy. Ultimately, we're going to probably have patients on cocktails of drugs, just like in cancer and diabetes and hypertension and COPD. Patients are on 3-4 drugs at a time because you can't just target one node in the cell that's affected by the disease because multiple nodes in the cell are affected by the disease. So we have to chip away at this. And the way we're going to define these precision medicine subtypes of patients that are going to be eligible for clinical trials and ultimately directed as part of this combination therapy approach that's going to happen is because of biomarkers. So I think it's a really exciting time in our field because we're breaking out. And I might add that of the over 100 drugs that are currently in clinical trials for Alzheimer's, less than half of them are against beta amyloid, and more than half of them are on novel targets. And so that's a pretty exciting consideration when we think about the fact that probably 30 years ago, which is not a long time frame in our space because the research only started around 1980, which is when I started, and we knew nothing literally about the disease. There were no drugs on the market and no drugs in clinical trials. So I think we're going to see a rapid increase in novelty. We're seeing companies like Denali and Elector and a whole spate of companies out there now going after novel targets, and I think they're relevant. And so it's very exciting. We've learned how to do clinical trials, then we're entering into an era where we're going to have innovation on targets.
Yeah, that's great. And Karen, I want to put this to you, which is, and it's one of the sub-bullets in the slide here, which is, well, it's actually two of them. One is, what will it take for this space to heat up? Do we need a win that has broader implications than just itself. And what I mean by that, if you look at immuno-oncology and the success in terms of all the flurry of activity that came out of basically the success of anti-PD-1s, PD-L1s, CTLA-4 antibodies before that, which generated 20, 30, 40% overall response rates in what were previously regarded as salvage cases, end-stage cancer patients, heavily pretreated, metastatic. And now the field is forced to reckon, given that there's so many different combination approaches being introduced, many of which are not necessarily panning out. The field is being forced to reckon, the oncology field is being forced to reckon with patient selection. Here, in the case of Alzheimer's, it would have been nice I think, to have had aducanumab succeed, even if it provided marginal benefit, because it would have potentially provided a platform upon which other adjunctive approaches could be combined. And then we can start really getting into this question of patient selection. Without even anything on the board, we're forced to reckon with this question of patient selection, I think, at a time where there is nothing kind of more pleiotropic that's available to patients, that's available to the market to provide a foothold. So how does the field deal with that? And specifically, are there any approaches on the horizon, therapeutic approaches or therapeutics combined with diagnostics that could serve to really ignite the field?
So lots of great topics within that question. I also I will commiserate it is depressing that we didn't have a big win in this space. Even if it was a modest benefit of aducanumab, I think that would have certainly been a nice important tide for this field. So that being said, here's the reality that we're in. How do you move from here? You as you look at the landscape, you can see how a number of people are moving, a number of companies and a number of investigators are moving to genetic subsets of neurodegeneration, saying, well, Alzheimer's, that's really hard. I perhaps can find a foothold into an overall broader neurodegeneration if I look at genetically defined subsets of ALS, of other forms of cognitive neurodegenerative diseases like frontotemporal dementia, progranulin FTD or versus tau FTD. So there are that is one direction that companies and investigators are moving into. Is so let me try to pick off what I can better understand the pathobiology potentially, and at least have some more homogeneous data patient sets with those approaches. In terms of and that is where I think the investor sentiment has moved towards, or there's already significant sentiment around rare disease formats, and I think that is something that these genetic subsets of neurodegeneration fit into those buckets, and seeing quite a bit of activity in that area. There's also the hope that if you can go after some of these genetic subsets that might be lower risk, and have that probability of success of more rare diseases that are precedented, that there may be some connection to the broader set of neurodegenerative diseases to Alzheimer's disease. So there's a company in your slide set that Blackstone's been involved with, where there's an approach of looking at both a subset of neurodegenerative diseases, but then also explore more broadly neurodegenerative Alzheimer's disease potentially. The other another point that I just wanted to comment on, and maybe more emphasize a point that Howard made, which is the criticality of biomarkers, the criticality of being able to do efficient clinical development. That is what our colleagues in oncology have, where you can do fairly cost-efficient phase 1, phase 2 studies that allow you to get quick signals as to whether your mechanism is relevant, and that barrier to entry for companies, for innovators, is something that we need to lower, and so the great work that's happening at whether that be the foundation level, the government level, to understand now how can we find patient subsets, how can we be able to study can we get trial ready cohorts to be able to move quickly through and cost efficiently through clinical studies, will be very important for us to have more and more approaches to trying to solve this tough problem of Alzheimer's disease.
Right. No, I think that's right, and the ability to understand which programs to kill versus advance relatively early in the development pathway would be a huge boon to the field. And in talking with Ken on a pre-call, he said the percent of phase 2 trials that have translated into phase 3 success in this field is 0%, right?
Yeah, asymptotic towards zero. Yeah.
So that's a real problem that the field needs to reckon with. And Ken, I'll put it to you, even though I'm sure you're going to be biased. What are some of the different therapeutic approaches that are out there, and you can include yours in the list, that you think are true contenders, both in terms of the modality being brought to bear, but also the way the companies are thinking about how to develop them in the clinic in such a way that they could actually be de-risked in a way that ultimately translates into clinical, regulatory, and commercial success?
Well, again, I'll go back to the fact that I don't know as much as my colleagues on the phone in terms of the specifics of each of the companies, and they may be better positioned to answer the question. I think the broad question that I deal with, which is slightly different, is is it a problem of bad drugs or bad endpoints to date? And nobody's really cracked this nut of correlation between phase 2 and phase 3. So there are people looking, downstream at other factors, at tau and glia cells and inflammation. And I don't think that any of us are in the position to say that any of those are, quote, winners from a scientific perspective, because I think there was a lot of belief in winners in the past. And again, it may be those were, the wrong drugs, and they're good endpoints, because we do have drugs that did get approval based on the existing cognitive endpoints. And it could be that they're bad endpoints and good drugs. And we're not going to know that until the trials are run. So I can't really speculate on what are the best approaches right now from that perspective, just because I'm scarred from the history that exists in the space. I do want to add that I think that we all want aducanumab to be successful, because the patients need it, and that's the most important thing. But I want to remind everybody that the difference between the oncology field and the Alzheimer's field is we're a pretty homogeneous patient population right now, and cancer is really a series of rare diseases. So each new drug helps individually, but you can find other places to develop new drugs. My big concern with aducanumab was on the rather interesting complexity that if that drug or any monoclonal gets approved and charges a substantial amount, the already extraordinary costs of clinical trial development in the Alzheimer's space might be to the point of really blocking out new drug development, especially by smaller companies. It's a whole different discussion of the economics of drug development in AD, but I don't think we can have any scientific discussion without looking at the entire clinical development pathway. So I'll stop there, and again, the scientific perspective may come from Karen or Howard in terms of detailed looks at other companies.
Yeah, I will put the same question to Howard. I want to come back to your question about economic burden But while we're on this topic, Howard, what are your thoughts about next waves of innovation? Where is ADDF looking? Where are you specifically? What things excite you about different companies, different programs, different aspects of biology that are being unraveled?
Yeah, well, we fund all over the world, and we're currently funding probably about 20-25 early stage biotech companies with innovative programs, all non-amyloid and frankly non-tau programs as well. I would categorize our main targets of interest or pathways of interest to be epigenetics, because epigenetics is known to be part of aging and plays a role in brain aging, and there's lots of evidence, and we're supporting a company called Horizon out of Barcelona, currently on the Madrid exchange for a phase 2 trial of a lysine-specific demethylase inhibitor. We are supporting a drug that is neuroprotective and cognitive enhancing that did have a successful phase 2 trial and is now probably the only drug in phase 3 for Alzheimer's that is a non-amyloid approach, and that's the drug from Aging Bio. Aging Bio raises the issue, actually, which is, I think, interesting discussion of the role of repurposing in Alzheimer's therapeutic development, and we could talk about that separately because I'm very excited about it. But Aging Bio is repurposing a very low dose of a drug called levetiracetam for patients with MCI. We're looking a lot at neuronal energy failure because we know the brain is, although only 3% of the body weight, it consumes 25% of the body's energy at any given moment from oxygen and glucose. And we know there's insulin resistance in the aging brain. We know that diabetes is a risk factor for Alzheimer's disease, and that patients with diabetes develop cognitive decline and impairment. And so we're repurposing some drugs like liraglutide at the Imperial College of London. We're in a phase 2, and we're thinking about a phase 3 program there in collaboration with Novo Nordisk, General Electric, and the Medical Research Council of the UK, as well as the Alzheimer's Society of the UK. We have a pretty big portfolio, actually, of drugs that try to improve mitochondrial failure and insulin resistance in diabetes. Inflammation, as has been mentioned, is probably the largest part of our portfolio. And we have a number of small companies that we've been supporting with anti-inflammatory drugs. Misfolding, I mentioned, we're conducting a clinical trial of a cancer drug that we're repurposing that attacks autophagy in cancer and probably plays might play a role in the treatment of Alzheimer's disease and other studies like that. And finally, I'll talk about the fact that there's so many ways that neurons get injured. Why not take advantage of the evolutionary advances in these cells? If you think about it, neurons are the only cells in the body that are laid down at birth, and they're still in your brain when you're 80, because when you learn to tie your shoes, those circuits were laid down, and those cells somehow have an amazing capacity to survive over the years, which other cells which are turning over don't. And I think by taking advantage of the evolutionary pathways for neuroprotection, namely mediated through BDNF and NGF, if we can drug those targets, that would be a big advance in our field because there's so many forms of injury. Again, we need that parsimonious pleiotropic effects. And we helped to start a company that's now spun out of, University of North Carolina originally, but now being managed out of Stanford, called Pharmatropics, that's in phase 2 with a P75 ligand that hits the NGF receptor that we're very excited about. And if those results come through, I think it'll be a big breakthrough. So that's a really brief aspect of our portfolio. We have over 100 active drug programs in 18 countries.
That's great. That's great, Howard. I'm going to put the same question to Kieran. I'm cognizant that we only have a few minutes left, but Kieran, as an investor, as a venture investor, with presumably a shorter runway to value inflection in order to generate returns, how do you think about innovation in the space and things that presumably need to get people very, very excited about a program in a limited duration of time? you can correct me if I'm wrong about that restriction, but assuming that's the case, then what are some of the innovations in terms of therapeutic approaches that are catching your attention?
Sure. Yeah. I mean, I think your overall statement on looking for value creation in a relatively short period of time, and that's in the 3 to 5 years is where many investors land. Of course, there are different investor groups that extend that runway, and there's other investor groups like public investors that shorten that and are looking for less than 3 years. So it is this is a field where you are not going to get broad investor interest just because we are talking about timelines that are likely longer to value creation. However, given the magnitude of unmet need, if there is strong pharma sentiment and continued interest amongst pharma on novel approaches to Alzheimer's disease, to neurodegeneration, that's not a beta, then you're going to get good, strong investor activity in novel mechanisms. And because the investment thesis there is, bring something to an interesting early clinical endpoint, or even just getting into phase 1. And because that mechanism is so exciting and interesting, that the pharma company will then acquire it and take that next risk level. So there's just overall laying out, the investment thesis will be different for different investors, but and we've seen that those investors that were early in tau, they did quite well. They exited fairly early because there was enthusiasm amongst large pharma companies for tau assets. So that whether that investment thesis will continue to hold, if you find the next amazing neuroinflammation target, which I agree is a pretty exciting area, and it's an exciting area because of some of the emerging human genetics. It's exciting because it potentially provides a thesis for being therapeutically relevant when patients already have symptoms. That's I think what has generated some enthusiasm around that the overall target space of inflammation. But where you need to as you think about the types of investors that are interested in this overall area, you've got to think through where the timelines make sense for their overall investment thesis. As I mentioned earlier in the call, you're looking at those more homogeneous genetic subsets is a direction where more and more companies have gone, because you can see how your project could get to a clinical inflection point, at least in the genetic subset, within that 3 to 5 year time horizon, even if you are not successful in the broader neurodegenerative disease. Just to add a few more points on what's interesting, to at least to me, in terms of emerging target classes, I think it's largely overlapping with a lot of what Howard is investing in, which is encouraging. We are also very much interested in neuroinflammatory targets, fairly broadly, whether those be glia specific or looking at cytokine-based approaches. I'm very also interested in a concept that is described as cellular senescence. And that's been interesting to a broad set of therapeutic areas, but certainly I think relevant and to this specific area of neurodegeneration. And then finally, understanding how to better approach protein aggregates, how to overall deal with what are many proteins that are likely causing cellular dysfunction. What are the best ways that we can reactivate our normal, our healthy systems like ubiquitin ligase-based proteasome systems or lysosomal systems. Those are areas where I think there's quite a bit of interesting approaches out there now. Those are activities actually also occurring in cancer, where we're seeing more efforts to try to activate proteasome pathways, and I think that work could be quite relevant to efforts in neurodegeneration as well.
Okay, great. And I was mistaken, actually. We do have till the top of the hour, so we have another 13 minutes to go.
By the way, I just might add on top of all of that, the one thing we don't know is whether or not all of these wonderful things have the right endpoint. And that I think is the underlying complexity of this field. Until somebody, to go back to the aducanumab story, but look at it a different way, in the last 15 years, there has not been a success with the current endpoints. And that's where I think there's pressure to think about that particular strategy. I think you may ask that question later, but I just wanted to, the science is all wonderful. The question is, how do you translate it into a drug?
If I may ask, Ken, do you mean clinical endpoints or biological endpoints?
Clinical endpoints that the FDA can accept.
Well, but Ken, don't you think that there's been so much work about cognitive scales, and the FDA is actually quite flexible on which cognitive scales we use, and there's a whole variety of them for the various stages of the disease. Now, my view on what you just said, if I may say so, as a clinician is, show me a drug that works, and I'm sure it'll move the cognitive scales. I think the idea of trying to get tweak one of these scales in a way that's not clinically meaningful makes it a difficult endpoint. But I think if we have a drug that has, for example, holding us to the fire as far as having a drug that's for disease modification slows the rate of decline by 30% over 18 months, that would be clinically meaningful. And if we had a drug that did that, I mean, you could probably just about use any scale you want to measure cognition and you'd find it.
And I just, this is not a debate for today, but it's a question of, I agree with everything you just said. I just don't understand why everything for the last 15 years, I don't believe everybody missed the question. It's why didn't it work? Why do we have 15 years where drugs didn't hit these endpoints and correlate between phase 2 and phase 3? Is it bad drugs or bad endpoints? That's just the question. It's not for today. It's going to be something that evolves over time.
Well, if I could respond to that, this has been written up in the literature, but one of the problems in our field has been exactly what you said, which is there's been a lot of phase 2 studies that have been underpowered and have not had biomarkers to show target engagement. So a lot of drugs, where there's a rush to the unmet medical need, which is so great, there was also a rush to go from phase 2 to phase 3, even though the phase 2 studies weren't done properly. They didn't have the clinical data. They didn't have evidence of target engagement. There was a signal somewhere, and there was a rush to phase 3. It's kind of like what Biogen did by going from a phase 1B to a phase 3 program. So I agree with what you're saying, but I think that now we're in a little bit of a different world where we're progressing to the point where I think we have better biomarkers and a recognition that we need target engagement in phase 2, and we need a clinical signal in phase 2 before we spend that $400 million or $1 billion on the phase 3 program.
And you and I agree completely.
Yeah.
And Ken, if I can interject, how I'm sure you speak regularly to regulators. How are they looking at things? Are they being proactive and innovative, or is it the companies that are kind of leading those discussions?
Well, I'll answer it in the following way. Since we're a private company, I can say this: we are moving towards a Type C meeting in the next few months. On the other side of that, I'll give you the answer. I think that I think the evidence suggests from other companies I've talked with that, I mean, the FDA understands the problem, and the problem which we're not going to again solve on this call is what's the best way to approach Alzheimer's, and how do small companies participate in the space? The economics moving towards a point where it's becoming impossible to think about any company moving all the way through to phase 3 and actually to marketing unless we can segment this market by looking at it in a genetically defined or cancer type of model. But the field is at risk right now for depressing investment from the venture community because of the alternative investments they can make that are positive. And so NIA has stepped into the void, the ADDF has stepped into the void, a number of other foundations have stepped into the void. But it's pressure on the regulators. They understand this. How they handle it, that's the unknown. Talk to me in 2 months.
And I'd like to add, this is Howard again. I've spoken to Billy Dunn, who I think is a very progressive head of the neurology group at FDA. And he understands the disease, and I've been on a number of panels with him. But, the bottom line is what I've heard from FDA is show us a drug that works and we'll approve it. And I know that's simplistic, but we really haven't had a drug that meets that 30% criteria that I mentioned earlier, which is accepted by the clinical world, by the clinical trial world. And basically FDA, they have parents with Alzheimer's disease too. And the guidelines that they wrote were very progressive. So I think FDA is very much on board with wanting to come to market with something that works, and they just want to see a drug that works and is safe, of course.
Yep.
Yeah, I would agree that the more recent guidance was from the FDA was quite progressive and progressive to the degree that I actually had some concerns that even a drug that was approved may not be paid for because that is not the discussion around being able to demonstrate clinical meaningfulness is going to be important for this field, certainly given the magnitude of the economic impact an approved drug would have.
Yeah. All right, good. So we have 7 minutes left. I have some great questions coming in from our audience. Karen, I'm going to give you the option of which of these you would rather answer, or maybe you can speak to both briefly. One is a question around novel mechanisms and aspects of biology that we haven't touched upon, including regeneration of neurons, the role of infectious disease, and companies like Cortexime, investors in biomarkers. Who are the big investors in biomarkers? And then there's also a question about whether VC activity has gone up or gone down in the last month or so. And I guess not much necessarily happens in terms of deal flow, but at least in terms of enthusiasm in kind of the post-Biogen announcement about aducanumab. Karen, do you want to take that for either of those?
Sure, I can take. I can give some brief bullets on several of those. In terms of of innovative approaches we haven't touched on, there are certainly more and more enthusiasm around gene therapy approaches. There more understanding around how to reduce that to practice. So we are there again for more genetic subsets. I do see quite a significant amount of effort around gene therapy based approaches. We're still hopeful that one of the more promising areas for cell therapy, I believe, could be Parkinson's disease. So I do think there might be some potential there in the near term for seeing some actionable results from cell therapy based approaches. I haven't spent much time thinking about infectious disease based mechanisms for Alzheimer's disease. I know that is certainly in the literature, and it's quite intriguing. At this point, I don't know how anyone can say that approach wouldn't work when we don't know what would work. So I'm quite open-minded to thinking about infectious disease-based approaches and mechanisms to Alzheimer's disease. With regard to overall investor sentiment, well, it could be better. Like, just to put it bluntly, that that if you had better data in Alzheimer's disease, I think you would get some more. You get more pools of capital interested in investing in Alzheimer's disease as follow-ons to seeing a path to success. And that's just that is as it is. However, the investor pool that is focused on high innovation, high unmet need is not going to be disappointed by an aducanumab failure. Is not going to stop putting capital to work in that area. in the area of Alzheimer's disease, as long as it understands, well, what's going to be different here? How are we going to address some of the fundamental issues that have been laid out on this call around better biomarkers, better diagnostic approaches, homogeneous patient populations, ability to get to more endpoints in an economically feasible way, in a capital efficient way. Those investors will still be interested in that. I haven't heard from my colleagues that they're not, no longer going to be working on Alzheimer's disease because of aducanumab's failure.
Yeah. Okay. Howard, any final thoughts from you? I know we only have a couple of minutes left.
Well, I've been doing this about 40 years. And again, as I said earlier, we had nothing 40 years ago. There weren't any papers coming out. Now there's thousands of papers a month. And I think we've learned a tremendous amount about the pathobiology of Alzheimer's. And now is an exciting time to translate that knowledge. And we're seeing increasing number of drugs go into clinical trials. And the science of clinical trials for Alzheimer's has advanced tremendously. And biomarkers, we've got innumerable biomarkers in the pipeline that are relevant and do all the good things that we've talked about today. So I'm very excited about where we are today. I realize there have been failures, but I think we learn from failures, and I think we've learned a tremendous amount from the failures that the focus on the amyloid hypothesis has garnered. Although it may turn out still, and I hope it does. So I'm very enthusiastic. And as far as the investor model, I'll just say one more thing. I think the trend in big pharma towards moving from internal R&D to external R&D has also affected Alzheimer's disease, but in a good way. And Pfizer is a great example, where they closed down their CNS internally, but they invested in $600 million in a venture fund that is going to be investing in earlier stage preclinical companies in our space. And I totally understand why VCs might not have the time frame to do this, but there's a lot of interest, and we're seeing it in pre-competitive consortiums. We're seeing it in alliances and partnerships like the Dementia Discovery Fund that was started by the UK and involved several pharma companies and venture funds like Gates Ventures. So from my perspective of 40 years, this is a really exciting time in this space.
That's awesome. Very, very optimistic. And Ken, we have maybe 1 minute left if you want to finish with any final thoughts before we just close out with housekeeping.
I'll finish where I started. There is clear evidence there's no loss of interest in the Alzheimer's space. There's a tremendous amount of diversity, development, and interest. And this is a societal tsunami. Nobody's missing that point. We've got dramatic developments in biomarkers. We've got progress towards what might be many polypharmacy approach to this disease state. So I would agree with my colleagues on the call that it's an exciting time. We need some wins, and clearly people are not going to stop because this disease is not going to stop. So, from all of those different perspectives, there's just a huge amount going on. It's going to be an extraordinarily exciting time. And we haven't really talked about the NIA putting a couple of $1 billion into the system as well on top of that. And that type of funding, for small companies such as ours and many other companies has been an extraordinary help during the gap, the traditional void that exists in funding. So there are a lot of resources focused on making this happen.
That's great. I'd like to thank all of you, Joel, Howard, Ken, and Kieran. It was a great discussion. We have lots of follow-up questions, so we will be following up offline for those of you whose questions did not get answered. And we will have the slides available on our website, and we'll send you a notice about that in the next couple of days. And thank you, and please feel free to reach out to us if we can be of any further assistance to anyone. Have a good day or evening. Bye.